So we're going to go on to the second paper from SSAT, and this is Dr. Rui-Min Mao, and the paper is entitled Therapeutic Effects of Oridonin Against Inflammatory Bowel Disease Involves Inhibition of Intestinal Fibrosis via NF-κB and P38/STAT1. And please present the paper.
So, hello everyone. My name is Diana Mao. I'm a resident at the University of Texas Medical Branch. I would like to thank GlobalCast MD and the SSAT for the opportunity to share our project. The therapeutic effect of Oridonin against inflammatory bowel disease involves inhibition of intestinal fibrosis via NF-κB and P38/STAT1 pathways. I have nothing to disclose. So inflammatory bowel disease, or IBD, affects approximately 3 million adults in the United States, and it has significant costs for both patients and society. Annual medical expenses for a patient with IBD is estimated to be $15,000, and the value of work-related opportunity loss has been reported to be up to $5.5 billion. Intestinal fibrosis is a common sequela of IBD. It's found more frequently in Crohn's, affecting 30 to 50% of those patients, but is also seen with ulcerative colitis, although less frequently at a rate of 5%. The fibrosis process can lead to complications that present clinically in the form of strictures, obstruction, and fistulas. EMT, or epithelial mesenchymal transition, is a process that contributes to the development of fibrosis. To elaborate further on EMT, it is a process that occurs on the cellular level in response to inflammation, such as that present in IBD. It's defined by the loss of adherins, tight junctions, and cytoskeleton proteins that are normally found in epithelial cells. So epithelial markers such as E-cadherin and ZO1, or zona occludens 1, are lost. These cells, in fact, gain extracellular matrix and fibroblast morphology, as well as migratory capacity. Fibroblast markers such as alpha SMA or alpha smooth muscle actin and fibronectin also are increasingly expressed. Consequently, the intestinal barrier is compromised, and this damage results in greater permeability through the intestinal wall. Our compound of interest is oridonin, a terpenoid that's derived from the plant species Rhabdosia rubescens. This plant has been used for hundreds of years in Eastern medicine to treat common ailments such as cough, abdominal pain, and muscle aches. Research has demonstrated that this compound also has antioxidant, anti-tumor, and anti-inflammatory effects on various tissues. And it's thought that these effects are due to the inhibition of multiple signaling pathways, including NF-κB and P38. In the context of the gastrointestinal tract and IBD, oridonin has also been described to have therapeutic effects. In studies with in vitro experiments and animal models, oridonin has resulted in the slower disease progression and decreased levels of inflammatory cytokines and cells. However, the molecular mechanism by which this occurs remains largely unknown. We had two hypotheses with our study. First, we hypothesized that oridonin would attenuate inflammation-related EMT and fibrogenesis. And second, we hypothesized that this would occur through suppression of known pro-inflammatory pathways such as NF-κB and P38. To conduct our study, we used rat intestinal epithelial cells, and the cell line was IEC6. We pre-treated these cells with oridonin and then exposed them to LPS, or lipopolysaccharide, to trigger inflammation that was representative of that seen in IBD. Following this, Western blots and immunofluorescence assays were used to analyze specific proteins. Our results first demonstrated that oridonin inhibited LPS-induced fibrosis markers and restored ZO1 expression, which supports our first hypothesis. The Western blot and its densitometric analysis on the right show that exposure to LPS increases levels of alpha SMA, which is a marker of EMT and fibrosis. However, this effect was inhibited by pre-treatment with oridonin. The same phenomenon was observed with fibronectin, which is another marker of EMT and fibrosis. However, the opposite effect was seen with ZO1, which is a tight junction protein and normally highly expressed in epithelial cells. Exposure to LPS decreased ZO1 levels, and pretreatment with oridonin was able to attenuate that effect and restore ZO1 expression. Our second finding was that oridonin inhibited LPS-induced NF-kappa B activation. Prior research has demonstrated that NF-kappa B is a key pro-inflammatory pathway that regulates alpha SMA, which is how it ties into our study. So oridonin inhibited the activation of NF-kappa B by preventing the nuclear translocation of NF-kappa B P65, which is a representative subunit. This was seen with immunofluorescence staining, and with LPS, you can see that P65 has translocated to the cell nucleus. However, with oridonin pretreatment, this effect is diminished and P65 has a greater distribution within the cytoplasm. The same effect is seen on the Western blot, and on the two right columns, you can appreciate that following oridonin exposure, the signal of the nuclear components of P65 has been attenuated. We also involved TNF-alpha on these Western blots because it's a pro-inflammatory compound with known involvement in IBD and the NF-kappa B pathway. Oridonin's inhibition of alpha provides further support of its anti-inflammatory role within IBD. In addition to its effects on P65, we also found that oridonin inhibited the phosphorylation of IKK alpha beta and I-kappa B alpha and prevented total I-kappa B alpha degradation. As a brief overview, NF-kappa B is normally inhibited when bound by I-kappa B alpha. The phosphorylated IKK alpha beta in turn phosphorylates I-kappa B alpha, which releases NF-kappa B and allows it to translocate to the nucleus. Phosphorylated I-kappa B alpha then undergoes degradation. This Western blot demonstrates that following the administration of oridonin, there is a decrease in the phosphorylation of IKK alpha beta and I-kappa B alpha. Inhibiting the phosphorylation of I-kappa B alpha also increases the total expression and levels of I-kappa B alpha so that its expression is greater with oridonin. Our third finding was that oridonin inhibited the LPS-induced P38 and STAT1 pathway as well. This required a two-part experiment, the first of which involved the specific P38 inhibitor SB203580. This P38 inhibitor dose-dependently attenuated the LPS-induced alpha SMA expression, as seen on both the Western blot and densitometric analysis. Oridonin is involved because it inhibits LPS-induced STAT1 activity, which is a downstream factor of P38. Again, this is seen in both the Western blot and densitometric analysis. In conclusion, we found that oridonin inhibits EMT-related fibrosis and does so via inhibition of the NF-kappa B and P38 STAT1 pathways. Consequently, oridonin may be a promising drug candidate for IBD and specifically within the context of fibrosis. Thank you for your time and attention. This is my email address here. I am happy to hear any emails and answer any questions that you all may have. Thank you so much.
Thank you, Diana. I'd just like to ask you, where did you find oridonin? What is it? Where did you get it?
Yes, so it actually is derived from, China. It's used a lot in their Eastern medicine and has had therapeutic effects on common ailments and been used for hundreds of years. And so I guess in the Chinese literature, there's been a lot of interest as to why this has been utilized and why it's been, effective. And so after breaking down the actual plant, they found that the most active compound was oridonin. And from there, that sort of expanded to utility in other realms and then on other, disease processes, and eventually found its way to us, with our IBD and GI research.
Well, good. We're going back to basics again. I think, foxglove plant type stuff. Todd, you had a question?
Well, I think it points out a bigger point here in that, my mother-in-law is Chinese and always giving me Chinese medicines in our family, and we sometimes roll our eyes, and they would seem to work. The question is, we just haven't studied them.
Right.
So one at a time, we're going to have to do this. We're going to have to take these therapies, these holistic therapies that people claim work, and this is a big point. This is a big discovery for that reason. Not only could this be great for IBD, but we know people who have gone to get alternative medicine for IBD, and they claim it works better than any of the other medicines we have. So I think this is phenomenal work. I understood about 1% of it, but it was I mean, it's a big deal. So as someone who had IBD, I'm excited to see that we really don't have great therapies yet. So that's great.
All right, we're going to try to catch up. But Dr. Rosen, you have any pressing comments here?
I thought it was great. I agree with everything. Sometimes the answer isn't as complex as we think. So good for you for looking into it.
You can use it on your hernia patients. I'm sure every one of them is going to get Oridonin.
I try everything to not operate on those people.
Okay, very good. Thank you so much, Diana. Thank you. So we're going to now go to a poll for the SSAT papers. They were great papers. And let me just read them to you, and we'll have the poll. The first paper by Ryan Morgan was development of postoperative local tumors and distant metastasis relative to a low-fat diet. And the second paper by Dr. Mao, Diana Mao, was therapeutic effect of Oridonin. So please vote, and we'll have your results of the vote in just a moment.
And the vote's moving back and forth, so please vote now. The vote is going back and forth. It just got tie. So keep going, and it's moving a lot.